So again, my name is Thomas Farb, and I'm co-founder and CEO of Thrive Bioscience. And I've started 17 companies. I've had 7 unicorns, and I'm working hard to make Thrive Bioscience the eighth unicorn. So we found this area that was ignored. I like finding ignored areas because it means people have stopped thinking about it, especially if they say things like, oh, it's always been done that way. It means there's usually room for innovation. And cell culture, tissue culture is exactly that. It is the basis of almost all biomedical research, delivery of cell-based therapies, in vitro fertilization, and yet amazingly, it's still done manually, like the way it was 70 or so years ago. So what we're doing is basically bringing cell culture into this century. This is similar to a company that I started called Cytic, where we automated cytology labs, which have pap smears. 80% of the work they do is pap smears. Pap smears are the world's most widely given primary screening test. And prior to Cytic, they were every year because they were so inaccurate. And as a result of our automation, we had 90% market share. And they've become every 5 years instead of every year because they're so accurate. We're doing the same thing, but in cell culture. I would have started Thrive after Cytic if I had looked for other labs that are unautomated. But I decided to look at other screening tests that were problematic. And so I started a company called Exact Sciences, which has a replacement for colonoscopy. Marty, next slide. So we're providing live cell imaging, analytics, and automation for mostly drug discovery, cell therapeutics, and bioprocessing. Next slide. So that's an image of the first cell line factory 70 years ago. And cell culture today is still similar to that. As one of our customers said, cell culture may be the only remaining island of outdated manual processes surrounded by an ocean of automation. What we're doing is basically industrializing this field. And to industrialize it, Marty mentioned my background is in several different areas. Well, that's what it takes to solve big problems. And so we're combining not only cell biology, image processing, machine learning, mechanical engineering, electrical engineering, optical engineering, databases, and others. And it's very difficult to keep this group talking to each other is a real challenge. So we are doing in a way what sequencing did for genomics. We're doing that for cell biology, which is lots and lots of reproducible data. So there's an image of that would be difficult to have done without Thrive, because we take images at 100 focal planes, and that's an image of a 3-dimensional organoid, which is the fastest growing area of cell culture because these organoids are closer to humans than just cells on the bottom of the plate. So we sell instruments and software that provide imaging, data, analytics, and automation. And we're basically improving cell culture, stem cell culture, drug discovery, the delivery of cell therapeutics, and ultimately bioprocessing. So it's easier for me to have a customer tell you about what we do than for me. So one of our customers, and I'll show you images from his research later related to COVID-19, described the current market as current tools are failing us because they hurt the cells. They only capture 2D single point in time images and don't capture contextual data or enough data. After using our instrument, he said, Thrive will change the world. Thrive allows us to understand and see living, dynamic cells and tissues in 3D over time with remarkable images. We hear a lot. We're able to see things we were not able to see before. So some background on the company. We started in 2014. We have 24 employees. 8 of them are on the sales and marketing side. About 9 months ago, we started selling our product line. And in the last 9 months, we've had orders for $13 million of products. 40% of our revenues are from software. And so that represents about 65 instruments. So we're projecting about $10 million in sales over the next 12 months and growing to 165 million in 5 years. And I've done that before. Basically, we put ourselves in a position where you have to buy our instruments, license our IP, or buy us. We have a very strong IP portfolio because there's very little innovation in this field. These instruments had not been developed. So we have 94 patent applications, 36 of which have been issued. We've raised $36 million to date, mostly from family offices. The S.A. Lauder family is our largest investor. They invested in 2 of my prior unicorns. We are just closing a $12.5 million convertible Series B convertible note. If you were able to make a really quick decision, you might be able to get into that. Our next financing is a Series B. We'll be doing that for 20 million or so at the beginning of this coming year. And I am smart enough to know that investors need a return. I'm a large investor in the company, about $4 million of the 36. And so it's nice that we fit well into 20 or so companies. All of the research tools companies such as Thermo, PerkinElmer, Agilent, the imaging companies, the bioprocessing companies as well. Next slide, please. So our business strategy is to build another unicorn, and the key elements of our business plan are find an important ignored area, provide full solutions. Big problems usually need full solutions, not a single point of the whole problem. We want to achieve and maintain our market dominance. I've had 2 companies that have achieved 90% market share, the Pap smear company, and actually your credit cards and debit cards are monitored by my software, and Fair Isaac bought my company H&C Software. We are building obviously proprietary patented, as I mentioned, 94 patent applications. It takes a cross-disciplinary team to solve these problems, and that's something that bigger companies are not very good at. And obviously, you have to keep an eye on value and building value. Next slide. So cell culture, even though it is key to most areas of life sciences, it's conducted almost completely manually and without data. This is one of the few remaining undigitized areas in the field. And yet we take these cells that we know so little about. In fact, Uber knows more about us than we know about the cells that we're studying. And we take these and then we spend billions of dollars on them running assays, putting them into other instruments, running clinical trials on them. And this is what's called the reproducibility crisis. It leads to lack of comparability of the data. So one lab's data is not comparable to another's. Huge variability in quality. Mislabeling rates are astounding. This is from the NIH. 11% to 30% of all cell lines are mislabeled, which means that people think that they're studying liver cells and screening for liver cancer drugs, but they're actually doing it on prostate cells. And this is not atypical of a field that's not been automated. You have years and years of buildup of errors. And it's incredibly inefficient. Researchers spend hours per day looking through a microscope. They put the cells back into the incubator and almost no data is kept. And therefore, you can see that researcher in the slide shrugging. Generally, researchers don't know why an experiment worked or did not work because they're not collecting data. Next slide. So I mentioned the reproducibility crisis. Nature magazine did a survey of 1,500 scientists. The majority of them said that this is a significant crisis, which is actually pretty scary. And it means that basically data cannot be reproduced. Part of the reason I started this company was in 2013, Amgen came out with a study. They had spent 10 years, had up to 100 scientists trying to repeat 52 53 landmark oncology studies, and after all that effort, they were only able to repeat 6, and others have found the same situation. Next slide, please. So how do we help solve that reproducibility crisis? This is an example, a really simple example of how we do it. I mentioned the fact that so many cell lines are mislabeled. Well, every customer says, oh, we don't do that. And this is one of the world's leading research institutes. And sure enough, they started using our instrument and they found that, gee, there's some cells that are growing faster than the others. Why is that? And it was mislabeling. It's sometimes because an incubator was unplugged or they used the wrong reagent or an outdated reagent. Next slide. So I mentioned a lot of business plans that I see, and I invest as well, take credit for this huge market, in our case, $29 billion, but they only solve one piece of it, and you have to solve all the pieces. And so very early on, we step back and look at, in order to bring cell culture, tissue culture into this century, what did we have to do? So number one, you have to control the environment. Right now, cells are taken across multiple environments, create stress, genetic drift. You have to have documentation. This is a huge problem. And particularly in biology, you need this kind of contextual data, particularly if you're going to do data mining or AI. And you have to acquire rapidly huge data sets, which is what we do. We're really the only ones that provide our customers are taking terabytes of images per week. And we put them into a simple database to make them actionable. When we take these images at 100 focal planes, we're able to track a single cell over time. And we adjust so that all the data is comparable even across labs. You have to do this with automation. There's no other way to make sure you have that comparability. And obviously, you have to be able to handle huge amounts of data in populating a database. My co-founder is responsible for many of the sequencing instruments that are sold today, which is one of the earliest life science instruments that had to deal with enormous amounts of data. And then you have to make this data actionable and visible. Marty, next slide. Sorry, I'm not sure which one. Okay, so here's our product line. We are not a one product family. These are products that we are currently selling or have just announced and will be shipping shortly. So we sell 4 instruments. The amount of analytics and software that come with this product, as I mentioned, about 40% of our revenue is from software. We provide a documentation system, which is really changing the way cell culture is done. And on the software side, we've introduced a whole new way of counting cells. For anybody who's in life sciences research, they know the incredible importance of counting cells, yet current counting is off by 40 to 80%. It's because cells look alike and debris looks like a cell, and 2 cells kind of squished together look like one. Others try to outline the cells. We do that too, but we look for what cells have one of, which is a nucleus. Seems kind of obvious, but people haven't done it. And we build 3D images so we actually see the topology of a cell. And so we've been in some bake-offs by large pharma, and it's 99+% accurate. This means that a lot of research probably needs to be redone because we literally, the number of cells you have affects the results that you get. Next slide, please. Next slide, please.
We got about 8 minutes.
Okay. I'll need you to move through those slides with me quickly. So we are providing really the right data, enough data. Currently, researchers will take images of what they think is interesting. The problem with that is that's highly biased data. So we take images of all the cells in all the wells in the lab, and each of these researchers are actually building these very valuable databases, which we also have access to. Next slide, please. So we offer usually a decision requires many different levels of approval. And we provide benefits both for a cell manager, a cell lab manager, so better yields, higher quality. They can reduce their expenses by comparing which reagent is better. But the researchers in particular are able to use this data to have really breakthrough insights to ultimately to drive automation. Next slide, please. So this is probably one of the most important slides in the deck. And what's different about us is our ability to take practically 3D. We take hundreds of focal planes of images, and we're able to track a single cell over time. That was something that was wanted by, I mean, it was a dream for researchers to be able to study a cell, because biology is a process. Next slide, please. You have to fit into the workflows of a laboratory. And so we have several different instruments. This one, the CellAssist 50, combines our imager with a 50-plate incubator so that in particular, these time series can be done, and it can all be done remotely. Next slide, please. I mentioned documentation. As a Boston Research Hospital said, with CellAssist, barcoding and software, everything is documented. We no longer mix up plates, forget which reagents we used, guess at who did what. And unfortunately, that is the status of cell culture without our instruments. And this is part of how we maintain our dominant market share, because once a researcher collects all this data, it makes it less and less attractive for them to move. Next slide, please. We also bring to the cell culture lab best practice kinds of software. So everything is remotely available, everything is archived. Next slide. So here's some of our customers, Battelle, Harvard Stem Cell Institute, New England Biolabs, Thermo Fisher, Wake Forest Institute for Regenerative Medicine. And basically we're adding to our product line, we used to just do cells, now we do tissues. We used to just do cells, we also now do stem cells. And we're adding different kinds of lighting, and we're adding software that are extending the usefulness of our instrument to key workflows. Next slide, please. So here are some images. These are the most important areas where our instrument is used. I mentioned the upper left, organoids. Just to the right of that is following of a colony over time. And the importance of that is that you want to have what are called monoclonal colonies. The colonies tend to merge and separate, and that's not what you want. So the whole industry is looking for monoclonal colonies, but they can't actually figure out which are without our instrument. The bottom left is an example of drug screening, which is our most widely used use of the instrument. And what's amazing about that is, for the first time, this lab was able to compare 50 different drugs, and with metrics, as opposed to counting, which is very inaccurate, the number of cells that have died, or this one looks better than the other. They'd never seen curves like this. Next slide, please. So I'll skip through these quickly. This is organoids, showing you the top of the stack and the bottom. This is the application was screening wound healing drugs. This is from the researcher who said Thrive will change the world. On the bottom, you can see the last slide is stained, and this is the most widely used assay in infectious disease research. This is how we evaluate whether a virus affects cells or not, and how it does. And it takes usually 2 weeks to have an answer, because we wait for these holes to form. And without our instruments, researchers can't see what's going on. So they have no data, they stain in 10 days or 14 days. They've killed the cells by doing that, so they can't follow them. And so they haven't been able before now to track what's happening before. This is a whole new area of data, which is why our customers said that will change the world. Next slide, please. So I mentioned our IP portfolio. It's in all areas that are needed in our field: image processing, analytics, the hardware, the software. It includes security, machine learning. Next slide, please. We have a team of 24 people from very diverse fields. My co-founder is much smarter than me. He's well known in the industry of genomics, and after spending 25 years in that field producing data that really changed genomics, we visited a cell culture lab together, and he realized what cell biology needs is the same kind of data, which just hasn't been available before. So Illumina, for example, is a company that we look at as a comparable, which is a several billion-dollar company. Next slide, please. Okay. So our main sources of revenue are currently mostly research. There are three main markets: research, it's about a $7 billion market; medical applications, which we're getting pulled into more quickly than we expected. We didn't expect it at all for several years, but in vitro fertilization is a great use of our instrument; and bioprocessing, which is the largest market. We've also gotten requests for licensing of our patents, so that'll actually be kind of a fourth market for us. Next slide. Currently, you can see that the majority of our revenues, about 3/4, are from research, and 5 years from now, we project that it'll be about a half, and the main growth is from medical, and then ultimately bioprocessing is the largest market. So in conclusion, we think we have the team, we have the solutions to really a major problem. We're basically bringing huge amount of data to this undigitized arena, which is hard to find. And as some of our customers have said, things like, we see things we've never been able to see before. We're game changing for drug discovery. This is what cell culture and cell imaging should look like. So if we have time for questions, we'd love to.
We do.
We'd love to.
Let's have a couple minutes. Rinka, you had a question?
Yeah, we did not mention the word AI. Is AI involved in any of your software or products?
Yes, it is. But more importantly, nobody's going to be able to do AI unless they have the data sets.
Yeah.
And so that's our first focus. So our cell counting uses machine learning. The viral plaque assay, we're able to identify, not in 2 weeks, the effect of a virus, but in day 2 or 3 by essentially using machine learning. But we tend to take it step by step, and it's incredibly valuable to have the data and to have rights to it, which we continue to have.
One more question.
As it relates to cell gene therapy, are you also involved in delivering those to the right area?
Yeah. So by delivery,
you mentioned delivery,
right.
in the deck.
So for autologous cell therapies, where somebody's cells are taken and then modified and put back in, our instrument is being used. We didn't expect that for a couple of years, but a large pharma is in the process of implementing it across all of their clinical trial sites. And this is a big market because, unfortunately, cell therapy is based on cell culture, and it's done as poorly as the rest of cell culture. And it's a bottleneck. So this is a huge market. We view this as a bioprocessing market, kind of a combination of medical and bioprocessing. And we're getting pulled into it much more quickly, which is great.
Something that's really interesting about what Tom does is that when he first came in, he was talking about how he's actually structuring a process and creating methodology on top of it, which you really can't do without his technology, right, for this business. But the other thing also, and we point out about successful entrepreneurs, is that he's been able to innovate and kind of create these new unintended areas. The in vitro, I don't think you anticipated originally. I think that came out of left field entirely for you, but it's a big market for you.
Yeah, in vitro fertilization is done as poorly as other cell culture.
Right, and so it just is a matter of Tom's maturity as a guy who's had 7 unicorns and had 14 startups or something.
17.
17 startups.
Close enough.
So I mean, so he knew when to zig or zag. So next, one more question.
A lot of success in startups is about zigging and zagging.
Yeah,
It's a very
exactly, right.
technical term, but
I don't want to get too scientific.
it's a good term.
Last question?
Yeah, I was an oncologist in the West Coast, and I gather you're culturing benign cells, and he had difficulty culturing malignant cells, and you have to put someone a rush to do that. Where are you in that process?
So the largest use of our instrument is malignant cells. Basically, drug discovery, drug response is probably a third of the use of our instrument. So typical workflow is somebody will take cancerous cells from a patient and try different drugs on it to see how they respond. Or,
Chemo, you mean?
yeah, cytotoxic or chemo. That's a very frequent use of the instrument.
Fantastic.
Thank you.
So thanks so much, Tom.
Thank you.